Hello, everyone. Welcome to the second module of the Wetlab series. I hope you all learned something from the first module about immunohistochemistry. And today we will be taking the next step, I guess you could say, from immunohistochemistry. And we're going to go over confocal microscopy. So this is how from immunohistochemistry, from the first module, we learned how to stain and prepare tissues for imaging. And now I'm actually going to go into the basics of the imaging process. And in the next module, we will be talking about experimental models and overall learning about experimental design and how, what factors and considerations to take into account when you're planning to develop your own wet lab experiment in the future. But for today, let me just share the PowerPoint slides and we will get to it. All right, so, sorry. Okay, let me just go to the first slide. So yes, today we are going to talk about the basics of confocal imaging, and this is the second module. In case you missed it from the first module, my name is Foong. I'm a fifth year PhD student in the Molecular Engineering and Sciences Institute. And I work in the NANS lab, which I'm sure you have heard about from Holly, and I'm sure you also learned about through applying for the textile program. But if you ever have any questions about wet lab or research in general, any one of us, me, Holly, and Els, will be happy to answer you. So please go ahead and Slack us. So with that, I do want to skip to this first session part, which is an activity that I would like you to do for the pre-module. This is the stuff that you'll see in the Google Forms that I will be sharing and releasing with you all. But I think it's really important, just if you're new to imaging, just to explore what capabilities are available for microscopy. So there are a lot of different companies that sell microscopes, but one of the popular ones and the one that the NANS lab uses is Nikon. So Nikon is actually really nice because they actually have a lot of training modules on their own website where you can learn more about microscopy or you can get a feel for the instruments that they make, as well as learn about the fundamentals of microscopy. And there's a lot of different types of microscopy as well. So I recommend that you start off learning from this website and just going through a lot of the different modules, just playing around with it. So I will be adding this to the pre-module activities that you'll be doing, because I do think it is worth looking into. And then I also want to just, since this is asynchronous, we can't really review it in person, but I do want you to just keep in mind what you learned about immunohistochemistry from the first module, because understanding the basis of immunohistochemistry will really help you understand this module today. So just keep in mind what types of features of confocal imaging do you think is important to keep in mind when imaging cells? So when you're imaging something, think about what you think would be important to to optimize in a microscope, what you think are important parameters to keep in mind if you want to image something. So I'm going to start off by just going over what confocal microscopy is. And I do want to point out that there's a lot of different microscopy types, but the one that we use extensively in the NANS lab is confocal microscopy. So something important to point out here is that confocal microscopy relies on fluorescence to produce images. They rely on these fluorescent molecules, called fluorophores. And from the first module, you'll remember that the fluorophores are usually bound to the antibody, specifically the secondary antibody. And these molecules, these fluorophores, release light, and the ability to collect fluorescence from different planes of focus that you're interested in is really what makes confocal microscopy really powerful compared to other other microscopy techniques. So a microscopy technique that you might be more familiar with is light microscopy. So that's just using like a light microscope. You might have had it in like your high school or your elementary school or middle school or what have you. Yeah, this type of microscopy just uses light to be able to see things. However, with this type of microscopy, you're not really able to get a lot of resolution. But with confocal, you can actually choose which plane you want the light to, or choose the light, choose the plane of focus that you want to look into. So you have a lot of different options on higher resolution when you're using confocal compared to regular light microscopy that kind of just illuminates everything. And you can't really be specific about the plane that you want to image. Fluorescence is the emission of light by a substance that has absorbed light or other electromagnetic radiation. So it's a form of luminescence. Some of you may recognize this as the electromagnetic spectrum. And this is basically a range of energy that travels through space, and the energies associated, and the wavelengths associated with that energy. But for confocal, we are specifically interested in the range of visible light here, which is blown up. Visible light exists in the spectrum. You may know this as colors of the rainbow. And what this means is that each color does have a different wavelength associated with it. So shorter wavelengths are bluer. So here, for example, let me have the laser pointer on. Okay, so yeah, the shorter wavelengths show up as more blue in color, while longer wavelengths are redder in color. So when you're imaging or when you're performing immunohistochemistry, it's important to select antibodies that have fluorophores that fluoresce at different wavelengths. So you can differentiate and visualize different objects from one another. So if you remember back from one of the earlier pictures that I showed in the earlier module, there were cells that have multiple colors on the same image. And you can only get that if you make sure that your fluorophores fluoresce light in different colors. Because if they all fluoresce at the same wavelength, everything's going to be the same color, and you don't know which. Like if you're trying to stain for different proteins, you're not going to be able to tell the difference. So it's important when you're designing your experiment to choose fluorophores that have different wavelengths if you're interested in imaging multiple things on the same image. So as I mentioned, visible light does exist in an energy spectrum, and many molecules do have the ability to absorb certain wavelengths of light. For example, something that you may not know is that leaves actually appear green, not because they're actually green, but because the chlorophyll molecules in the leaf absorb wavelength of light in the blue and red spectrum. So yeah, the leaves, the chlorophyll absorbs light from the blue and red spectrum. So really what's all left is green. So after subtracting the blue and red light from sunlight, much of the visible light is green. Once absorbed, that light can then be converted into chemical energy through photosynthesis or into heat. So in this instance, with the leaves are actually absorbing absorbing the light, and then converting that into chemical energy. So it's converting it into photosynthesis or heat. Other molecules have the ability to re-emit light energy, and this process is called fluorescence. So that's what we're going to go over in the next slide. So as I mentioned earlier, first and foremost, confocal microscopy relies on fluorescence to produce images. So you can imagine that you have this light here, and you have this fluorophore, say, at the resting state. What we're going to do is you're going to shine light on the fluorophore, and what's going to happen is that you're going to excite that fluorophore up to a higher level of energy. So it's going to go up, and then what comes up must come down, right? So eventually the fluorophore is going to lose all that energy, and then go down back to the resting state. And at this point, as it goes down to the resting state, it releases energy in the form of fluorescence. So it re-emits that energy as fluorescence, and this is what we capture with the microscope and how we're able to visualize the proteins or the cells or whatever target we're interested in. So it's really important to choose fluorophores. Just as I mentioned, it's important to choose your antibody. Again, you have to make sure that different antibodies don't have the same fluorophore. That way you can image multiple targets at the same time at different colors. So it's really important to choose the right antibody, but it's also important to choose which type of fluorophore you want to use. Now, you might notice through just doing a lot of research or looking at cell images that a lot of cell images actually appear in red and green. And you might be wondering why. Well, the answer is that the emission spectra is actually spread farther apart, so it's easier to tell the difference between the two. So here you have the dotted line is the excitation, and the colored peak is the emission. So you can see here for the green, it's excited around like 480 nanometers, and it emits light at a peak of like 520, say, versus the red here, where the emission is more like 670, 680. So the peaks are different. And that's important because if you can imagine, if the peaks overlap, then whatever light you shine on it, both fluorophores are gonna fluoresce at the same time. And it's gonna be hard for you to determine the difference between the two. They're both gonna be like very similar colors, right? So it's important to choose colors that are farther away from each other. And this is why a lot of red and greens are used in imaging. Blue is also a popular color for the nuclear stain, the DAPI, like I mentioned. So yeah, you just want to make sure that when you're designing experiments to choose fluorophores that are farther apart from each other on the emission spectrum. And this is a really neat tool to be able to look at the different spectrums so you can really visualize where the peaks are. So while traditional fluorescence microscopy provides useful information about like the localization of fluorescent compounds, the images produced can look blurry or really out of focus. And this is because the emitted light that's collected from the image can originate basically from any plane. You really don't have a focus, as you can see here. You really don't really have that much resolution, that much focus, because what happens is that it's basically shining a bunch of light onto your sample. You're collecting all of that. So it's like too much light, and it's kind of really difficult to see details from one another. Now that's where confocal imaging can really shine. Confocal images are powerful because they can produce sharper images by using a laser to specifically excite target wavelength. So you can just see the difference between the fluorescence and the confocal images here. So for example, for our lab, we image a lot of tissues. And as you can imagine, tissues contain many, many layers of cells. They're just cells layered on top of each other. And if we use just a regular fluorescence, what's going to happen is that all the cells are just going to shine, fluoresce all at once. It's going to be really hard. It's going to be a lot of light there. So for confocal, you're shining a light, the laser, to excite, again, you're exciting the fluorophores with a narrow plane of focus. So you can control what plane, what layer of cell you're interested in imaging, rather than imaging the entire stack of cells in a tissue. And this is because there's a pinhole feature that allows only certain amounts of light to go through. You're not overwhelmed by so much light. So this pinhole really helps narrow down different planes that are fluorescing. So this is an example, and this is basically what happens in a confocal microscope if you were to look inside of it. A laser is used right here to provide light, excitation light. And this is really important to get really high intensity light. And it reflects off this dichroic mirror, which then this laser hits two mirrors, which are mounted on motors actually. And these mirrors, these objectives actually scan, they direct the light to scan the sample of interest. Dye in the sample fluoresces. So imagine that the sample was already pre-stained with antibodies and fluorophores. The dye in the sample fluoresces and the emitted light, this is green, this is the emission, the emitted light gets descanned by the same mirrors that were used to initially like bring light into the microscope. So basically the light goes back the same way. The emitted light passes through the dichroic and is then focused into this pinhole. So you have this focusing happening. And then as the light passes through the pinhole, it's captured by a detector, which then produces an image on your screen. So yeah, it's really this pinhole that really is a huge advantage of the confocal microscope compared to the fluorescence microscope, because it's with the pinhole feature that you're allowed to detect specific wavelengths of light. You can really control the plane at which you're imaging. So I will also upload a video of how confocal microscope is used in the lab. So I hope you enjoy it. Please let me know if you're not able to access it. But if you wanted to see what an actual confocal microscope looks like, or how to use it, or what it looks like or what it feels like to image a tissue slice in real time, please watch that video. I think you'll have a lot of fun with it. These are not videos, but these are just pictures of what the confocal looks like in our lab. It looks like honestly, if you've ever seen a fluorescent microscope, it's just a beefier version of the fluorescence microscope. It has a lot of different laser settings that a fluorescent microscope doesn't have, and it has that motor as well, as I mentioned. And you can't see it here, but there is like a small pinhole that you can control. And it's connected to to a computer that you can use to be able to capture the images and be able to display the tissue or the cell live on the screen. And this is a close-up of the different lens and the objective. So you have these binocular lens, which you're probably familiar with in a lot of different microscopes. But what's really unique here is that there's multiple objectives. You can kind of see it down here, but there's multiple objectives that give us different magnification. So the lowest one that we have is 10x magnification, but we can go up to 100x actually, which is really cool. And you can also, again, control the pinhole and everything from this station. And this stage right here, the square in the center of the microscope, that's where you actually can mount your slide. So there's this little like section here in the center that you can slide back and forth to adjust to the size of your tissue or cells or whatever sample you have. So it's really compatible. This kind of system is compatible with a lot of different samples and different sample sizes. Okay, so before you do any imaging work though, it's important to think carefully about the magnification that you want for your experiments. So for this, you want to consider a microscope of view. Now, the field of view, or the FOV, is the maximum area visible when you're looking through the microscope eyepiece. And it's usually quoted as like a diameter measurement. So the field of view decreases when the magnification is increased. So as you can imagine, you're zooming in, right? So as you're zooming in, you see less, the area that you're looking at is smaller, but at the same time, you have greater resolution. So for example, if you're viewing a coin and you want to zoom in to like 100x to more details, of course, you're going to see less surface. You're going to see lower surface area of the coin. So this is just a couple images of what different objectives look like. Again, our microscope, we can go up to 100x. We also have a 10x objective and a 40x and a 60x that's not represented here. But this just gives you an idea of like how zoomed in with the different objectives. And on top of these objectives, we can actually zoom in even further. So I've actually imaged things at 240x before. And I think it's really cool that there's these different settings that allow you to magnify your images. So let's think about what types of objectives. Again, my goal was just that you can have a better idea of the types of things that are important to keep in mind when you're creating your own experiment. So something that's important when it comes to imaging is figuring out which objective you would need to use. And again, it really depends on what you're trying to image. So for example, here's a picture of 10x. And this here is a half of a brain slice. So this is one hemisphere of the brain. I actually think this is the right hemisphere, but um, yeah, this is one half of the hemisphere of the brain. And as you can see, I've made sure that my fluorophores don't overlap each other in the emission, so you can clearly see each color differently. There's blue, which stands for DAPI, which is the nucleus, green, which stands for microglia, and then red, which stands for like dead cells, basically. So you can see that all the colors are very differentiable because I made sure to design my fluorophores or pick fluorophores that would work well. And a 10x is big enough to be able to measure like one hemisphere of the brain slice, which is actually quite small. I would say this is like maybe half an inch across in diameter right here. So that's what a 10x is capable of. 40x, at 40x, you can actually start seeing cells at this magnification. Here we have some microglia, so you can start seeing some microglia at this magnification. You can also see the different microglial shapes. Polly probably has talked to you a lot about how cool microglia are because they are very dynamic and they have a lot of cool shapes. So yeah, this is really cool. In this image, this is one that I took actually. I was interested in imaging quantum dots, which are basically nanocrystals that have really bright fluorescence. In this case, the quantum dots are fluorescing in red, but you can see the dots right here. That's where my quantum dots are located if you look closely. But yeah, so it's really cool to be able to see cells and what the cells are actually looking like in tissue. Oftentimes you can even tell whether or not a cell is healthy. So these cells are pretty healthy, but you can tell if the cell is apoptosing, it'll look like it's breaking apart. So yeah, at this magnification, it's really cool to be able to see the cells that you're actually working with. And even cooler, at a higher magnification, at 60th, you can zoom into a single cell, actually. You can zoom into a single cell, and here you can see very clearly all the quantum dots that are attached to this microglia here, right? From this, actually, the 60th picture is actually taken, zoomed in version of this right here, this box that I drew. So you can see that like the really bright red that matches this really bright spot here. And yeah, you wouldn't really be able to tell that there were so many red quantum dots if you looked from 40x. We could really tell closely at the 50x, and that's why that's really the power of being able to go up in magnification within a confocal. And as you can imagine, you can see here there's actually no background, right? You have the cells, which are in green, and there's also black. So it's really easy to determine what is a cell and what isn't. And this is because of that pinhole, if you remember when I talked about earlier. The pinhole allows you to be able to image just one plane of cells at a time. Because you can imagine, this is just one plane of cells. There's more cells underneath. These are like stacks of cells, right? But the way that I have it imaged right now, you don't see the stack of cell underneath. You don't see the layer of cell underneath. I only image the layer of cells of this plane, and the other planes don't get. You don't see the cells from the other levels. So that makes the images really clear and crisp. Otherwise, you're gonna just see cells everywhere, and it's not. Again, it's just like really, really low resolution. So that's the power of a confocal microscope. So you're near the end of the presentation. I just want you to know that by now you actually know a lot of important features of confocal microscopy for people who might not have ever imaged anything before, much less use a confocal microscope. I think you're pretty well equipped to at least understand how they work and why they're really powerful, which is the goal, honestly. Now, even though today I focus on the confocal microscope, I do want to point out that there's a lot of different types of microscopy. And the type that you'll use will really depend on your experiment. It will depend on the lab that you have. So I just wanted to go over some other common microscopy uh, features or types that you might encounter in your journey to science. But in addition to confocal microscopy, there's light and optical microscopy, and this is the most common type. This is a light microscope you're probably familiar with maybe from high school or middle school, but it's just like what you would think of as your average day-to-day microscope. And the light microscope uses light to magnify the image of the object. Basically, you try to pass light through it or reflecting light off of it and then examining this light through a lens. So there are a couple different types of optical microscopes. The confocal is actually one of them, and there's also fluorescence and bright field, for example. So yeah, I would say the light and optical microscopes are the most common, and each one of these, oh, I am biased. I think the confocal is probably one of the most powerful optical microscopes out there, but there's of course other ones depending on what you need to do. Sometimes a confocal microscope, like for some imaging, you don't need a confocal microscope. You can actually get what you need with just a plain fluorescence. So like if you're doing cell work, and you can imagine when you're growing cells on a plate, on a petri dish, there's only one layer of cells. You're not going to grow multiple layers. Like you're not going to grow like a tissue on a petri dish. So you're only going to have one layer of cells. And because you only have one layer, you're not worried about the background. Like you're not worried that, I'm going to go back to this image here. Yeah, here, right? You're not worried about the cells that might be hiding in different layers of the, like you would in the tissue, because there's literally only one layer for the cell culture. So because of that, you really don't need the pinhole. Like you don't really need the confocal, the pinhole that the confocal microscopy provides. So you could get away with just using a regular fluorescence microscope or even a light microscope if you want to image cells. So that's one example of, it really just depends on what you're imaging to be able to choose which microscope you use. Another type of microscope is the electron microscope. I think electron microscopes are really cool. It basically replaces traditional lenses with electromagnets, and it uses this electron beam to create an image. So the electron basically bounces off of the image and creates really sharp, contrasting black and white pictures. And there's two types of electron microscopes. There's transmission electron and scanning electron. Yeah, go ahead and look up pictures of them in your own free time. I think electron microscopy makes super, super cool images. It's probably my favorite out of any microscopy images. And lastly, there's scanning probe. And this one's actually really interesting. It's different from other microscopy techniques that you might have heard about because it involves the interaction of a scanning probe with the surface of the sample where images are produced. So with this type of microscopy technique, you're actually it's more, I think, appropriate to say that you're feeling the sample rather than seeing it. Because what happens is that you have a probe, imagine like there's a record player, right? And you have the what do you call it? The bar of the record player that goes down that touches the disc, right? It's kind of like that. You have a probe that actually touches your sample. It drags along the entire sample. And every time the sample has a bump, or every time there's some different topology on the sample, the little hand on the little probe will detect that bump and will be able to image that. So a scanning probe is a really, really great option if you're interested in like learning about the topological aspects of their sample. So the surface of the sample, trying to understand the texture, roughness of the sample, for instance. This will be able to detect it really well because you're literally taking a probe and just dragging it across your sample, and it will be able to detect microscopic changes in the terrain. So yeah, it's really cool. It also provides images that are very different from traditional microscopy images. So I encourage you to look up images from all three of these different types and comparing them and see how they're different actually, because it's really cool. So yes, that's the end of today's lesson. I think, as I mentioned earlier, I hope that I think you're actually pretty well equipped to understanding the basics of microscopy. There's a lot of resources out there, and I hope this gets you thinking more about like, if you're going to design an experiment, what type of objectives you use, what type of microscope you'll need for it, what colors you'd want to stain your samples at, for instance, to be able to, fluoresce and see multiple colors at once. So these are all really important things that you should think about when you're working on a research project. And so, yeah, I hope that you'll be able to get a chance to just explore a little bit of what exists right now of current like pictures and microscopy techniques from the microscopy techniques that those different images use. And really just start to think about whenever you see, in the future, whenever you read a paper with an image, start thinking about how that image was obtained, because it takes a lot of optimization and a lot of work. Yeah, all right. Well, this is the end of the second module. The last and final module will be about experimental models, but this is kind of taking a step back and really focusing on experimental design, specifically what type of model, whether you want it to be in vivo, ex vivo, in vitro, what type of model you want to use for your experiments. And yes, please remember to watch the video of the confocal microscope in action in the MANF lab as well, so you can just see what that experience is like. All right, see you in the next module. And I hope you all have a wonderful day.